Part 2, building the model, the easy parts. Now, in standard comparative modelling, the next step that we do is to identify the structurally conserved regions and the structurally variable regions. So in the general case, the reliable definition of these is only possible if we have multiple parents. Where there's only one parent, we assume that all regions are structurally conserved except for regions where there are insertions and deletions, and that's almost exclusively in loops. So where an indel occurs in a loop, the whole loop is defined as being a structurally variable region. So this is an example where we've got two structures overlapped, and you can see circled up here three structurally variable regions which are loops which have flipped in conformation or maybe got an insertion or deletion. So here I've taken those loops off and just left the structurally conserved regions behind. In antibodies, the problem is rather different and really quite trivial, because the frameworks which support the CDR loops are really highly structurally conserved. They very rarely have insertions and deletions, and when they do, those tend not to be anywhere near the kind of business end of the molecule, which is the hypervariable loops or CDRs. So the CDRs then are the structurally variable regions. They vary in conformation, they vary in length, and they vary significantly in sequence. So here we are again looking at an antigen's eye view of the FV fragment, and we have the 6 loops which come together to form an almost continuous surface with the framework supporting them behind in this picture. So we have the loops L1, L2, L3, H1, H2 and H3. The next step then is to build the structurally conserved regions. In the general case, we can model a domain based on a single template or on multiple templates. In the case of antibodies, what we generally do is just use one template for the VL domain, the light chain domain, and one for VH, the heavy chain domain. Again, there are different approaches to select which structurally conserved template we're going to use. We can screen the frameworks first on the basis of canonical loop conformations. So although we are looking at the structurally conserved regions, we can choose which is the best framework based on the loops that they support. And as I said, that's done with this idea of canonical loop conformations, and we will return to that in a couple of minutes. Secondly, we can choose one antibody which has the best sequence match to both VL and VH together. And thirdly, we can choose the VL and VH from separate templates. So typically that would be done by taking into account these canonicals, but it might just be done on the basis of sequence identity or sequence similarity. Now the frameworks of antibodies really are very highly conserved. So here I've overlapped a set of antibodies from both mice and humans, and you can see that the structure really is very similar. There is a fair spread across some of these regions, but that's largely as a result of variation in the packing between the VH and VL domains. Because if we just look at this heavy domain on the left by itself, then you can see that these regions really get much tighter. There are a few little regions where there's some variability. That's that extra loop, which is not one of the CDRs, but you can see there's some variability in the tip of that loop. Now I mentioned this variability in packing between the VH and VL domains, and this is something that we looked at in my group, and this shows you the extremes of the variation at the time that we did this. And there's approximately a 30-degree difference between this angle here and this angle here. And what we're doing here is choosing two conserved strands. So we're taking those conserved regions in the heavy chain and drawing a best fit line between them, and doing the same in the light chain, and then calculating an angle between these two lines. And of course, this implies that given that we have this amount of variation, we need to choose the correct orientation between the two domains. And there are various approaches to handle this. So if we're using the same parent for both VL and VH, then basically we don't worry about it. We just use the packing angle, as we call it, from that parent. If we're using different parents, then we can inherit the packing angle from one of them, or we can try to predict the packing angle directly. Now, if we're going to inherit from one of them, how are we going to do that? Well, this is actually conceptually rather simple, but quite difficult to explain. So suppose we have our target sequences. We have our light target and our heavy target. And we have the parents we're going to use. So there's the light and heavy of parent A and the light and heavy of parent B. And we're going to generate a model for the light and a model for the heavy, and then pack them together. So the question is, if we've built these models LM and HM, do we pack them based on this packing angle LAHA or this packing angle LBHB? So what we've chosen to do here is build the light chain target based on antibody A's light chain to build this model of the light chain, and we've chosen to build the heavy chain target based on antibody B, and the heavy chain of antibody B gives us this model of the heavy chain. So what we do then to select which packing angle to use is we look at the sequence identity between the light chain target and the light chain of B. In other words, the parent that we didn't use. And we also look at the sequence identity between the heavy chain target and the heavy chain of antibody A. Again, the chain that we didn't use in the modelling. If this red one has a higher sequence identity than the purple one, then we choose this packing angle. And if the purple sequence identity is better than the red sequence identity, then we choose this packing angle. So what we end up with then is, if this were the higher sequence identity, then we'd use this packing angle, and therefore we take our model of H, the heavy chain, and fit it onto heavy chain B, and we take our model of the light and we fit it onto light chain B, and that gives us the packing angle that we've inherited from there. If on the other hand it's the other way round and the heavy chain had a higher sequence identity than the light chain did, then we take our model of the light fitted onto light chain A and the model of heavy fitted onto heavy chain A, and that means we'll end up inheriting the packing angle from structure A. Okay, so we've looked at how we build what we might refer to as the easy parts, the frameworks. In the next part, we'll go on to look at how we build the difficult parts, in other words, the CDRs.